human brain development is a precisely orchestrated process requiring multiple genetic and epigenetic interactions and the coordination of cellular and molecular mechanisms, perturbation of which leads to a plethora of neurodevelopmental phenotypes depending on the spatial and temporal effect of the disturbance. This process requires a multitude of genes, many of which remain to be associated with human disease.
We're very excited about this work as it represents a significantly sized genomics study. Over 120 families where the program manifested brain malformation were studied by whole exome sequencing. Usually, if there were two affected members of the family, both had their sequence done. If it was just one, we would often do a trio with both unaffected parents to try to initially focus on new mutations or homozygous variants in suspected genes that might be involved.
Our approach of sequencing 2 or 3 affected members from small families with apparent recessive inheritance without prior homozygous mapping differentiates this study from classical studies of recessive pedigrees. We illustrate the utility of this approach and underscore the added benefits of solving blended phenotypes and observing the mutation load of individual cases within a given pedigree.
In the paper, as expected, we find a number of known genes, but given that mostly a Turkish population was studied, the variants are different. So these report novel variants in known genes. And some of the novel variants in known gene also led to a phenotype that was different than what was described before. This is called phenotypic expansion.
In our analysis, we tried to maximize the power of a large cohort. We identified multiple families with different variants in the same gene, but we're also interested in determining whether proteins encoded by candidate genes identified in patients with similar phenotypes interact. By building in silico interactomes or networks that included both known and candidate genes, we were able to add potential links between stages of brain development, molecular processes important during those stages, and resultant phenotypes.
We used a combined approach to rare variant detection in order to identify both copy number variations, CNVs, and single nucleotide variations, SNVs, thus maximizing rare variant gene discovery. This comprehensive approach allowed us to identify even small intragenic hemi and homozygous deletions, which are essential to maximizing disease gene identification in Mendelian disease cohorts.
One of the major points of this paper is in regards to how we perform CMV analysis from whole exome sequencing data. In addition to performing CMV analysis using Conifer algorithm to extract the relatively large deletions and duplications, we developed our own algorithm HMZ-DelFinder to detect small and intragenic deletions from whole exome sequencing data. At the first step of this algorithm, we extracted read count information from each sample's BAM files and then retrieved the exons with very low read count. By this way, we found 4 families with homozygous or hemizygous deletions detected by this algorithm. Interestingly, these deletions lie within the genes that are associated with the patient's presented phenotype.
The vast majority of the families presented in our study had image-confirmed structural brain malformations, nearly 80%. This discrepancy is noteworthy as imaging studies provide specific objective visual and quantitative evidence for the genomic findings in patients with various brain abnormalities. Similarly, the type of brain malformation in a given individual and then understanding of its underlying molecular pathogenesis were utilized in the prioritization of the potential candidate genes identified in this study, like kinesin 23 in primary microcephaly, in AGBL2 in tubulinopathy-related cortical dysplasia syndromes, and this will not have been possible in a cohort of non-syndromic intellectual disability, and distinguish this work from several other studies published before.
We're very excited to see this come out. It's a large body of work, and we think we're starting to get more insights into what are the genes important to the development and function